Good morning, friends. Today we had just discussed the one bit from your NEET syllabus, which is a part of two chapters, cell as well as respiration. So our bit is mitochondria. As you know, when we observe the plant cell or animal cell, there is a number of organelles present in it. Out of them, you just mark it or label it 3-4 times mitochondria. This is one of the important organelles and commonly it is known as powerhouse of the cell. So it is known as powerhouse of the cell. So mitochondria is called powerhouse of the cell. So definitely by this sentence we understand powerhouse. And what is power for the cell? That is ATP or biological energy. So your organelles, mitochondria associated with synthesis of ATP molecule. Synthesis of ATP molecule. As we know, ATP is chemical or bond energy. So it is chemical energy or bond energy. For the cell, ATP is called energy currency. Without it, ATP cell cannot perform any kind of metabolic process. So therefore, it is one of the important cell organelles. First of all, mitochondria is colorless. So study of the mitochondria is required is staining or biological coloring. So colorless है, so stain is required. The stain of mitochondria is genus green B. This is an organic stain which is used to coloring the mitochondria. After that staining or coloring, we can study the mitochondria. So first of all, morphology of mitochondria. Shape. Now it is clearly visible in electron microscope by the coloring shape of the mitochondria. Size of the mitochondria. Number of the mitochondria. Okay. So shape of the mitochondria given in an NCERT text that is sausage shape or what we get cylindrical and size, length and diameter. So length 1.0 to 4.1 μm, while its diameter is 0.1 to 2.0 μm. There is small changes given in books, but it is almost 1 to 4.1 μm is the length and the diameter is 0.1 to 2.0 μm. And number is always variable. But number is dependent on metabolic rate. For example, meristem cell of the plant and another permanent cell, that is parenchyma cell. So meristem cell is more metabolically active. Therefore, number of mitochondria is greater in meristem cell. So this is a morphological feature. Color. You observe this mitochondria after staining, colorless. The shape is cylindrical or sausage. Size along with the length and diameter. So it is one of the small organelles. And number is depend on metabolic rate. When we observe the diagram of mitochondria, कि mitochondria हमें कैसा दिखाई देगा? Before it, we just know the three important facts of the mitochondria. This is a more important hashtag point. Mitochondria are semi-autonomous cell organelles. Semi-autonomous. Because of DNA. Assertion reason. Why it is semi-autonomous? Because of the DNA present in mitochondria. तो इसके पास जो हमें मिलेगा वो होगा डीएनए का मॉलिक्यूल। नाउ नेक्स्ट पॉइंट, mitochondria take part in a specific kind of inheritance which is known as maternal inheritance. So mitochondria take part in extra chromosomal inheritance. It take part in extra chromosomal inheritance. Another important point related with the mitochondria is mitochondria show the biogenesis. आपकी टेक्स्ट बुक में जब आप लास्ट लाइन पढ़ेंगे माइटोकॉन्ड्रिया की सेल में, तो वहां पर क्लियरली लिखा हुआ है इट इज डिवाइड बाय बाइनरी फिजन एंड दिस प्रोसेस इज कॉल्ड बायोजेनेसिस। तो माइटोकॉन्ड्रिया अपने ही जैसे माइटोकॉन्ड्रिया इजीली बना सकती है। सो बायोजेनेसिस बाय फिजन, दिस इज द जनरल इंफॉर्मेशन अबाउट माइटोकॉन्ड्रिया। सो semi-autonomous organelles take part in extra chromosomal inheritance, and one of the most important feature, it show the biogenesis by fission. So one mitochondria form two daughter mitochondria like your cell. So these are the three important hashtag point. Remember it. These are the specific feature not present in all cell organelles. It is present in mitochondria. The next focusing point is structure of mitochondria. What are the different kind of particles, enzymes present in the mitochondria to perform the ATP formation or aerobic respiration. कि aerobic respiration या ATP formation में mitochondria काम कैसे करता है? Mitochondria में अब हम पढ़ रहे हैं structure of mitochondria. So mitochondria is just a cylindrical shape. So this is the first part of the mitochondria called outer unit membrane, and another part which is showing this type of projections. This is inner membrane. So there are two unit membrane of the mitochondria, outer and inner. So this is called outer unit membrane of the mitochondria, and this one is called inner unit membrane of mitochondria. Okay, and the space between two unit membrane is outer chamber of mitochondria. Okay, and the space within an inner membrane is called inner chamber of mitochondria, or sometimes we can say the central cavity, inner chamber of central cavity. And the inner chamber of the mitochondria is filled with matrix. By observing this diagram, we can observe this one part, but when you just focusing the one part, another area is appear to you. Suppose this is an outer unit membrane, okay, and this one is folded is inner unit membrane. So this folding of this inner membrane is called cristae or crista, and this folding part bearing a small stalked particles like this. This folded part bearing a small stalked particle which are observed by a scientist. This one. So these small stalked particle on an inner membrane is labeled as oxysome. So technically, mitochondria having two unit membrane, two chamber along with matrix. Two unit membrane, two chambers along with matrix, and the two structural component of it is cristae and oxysome. So these are the component of mitochondria. Now we learn the characteristics one by one. Okay. So first of all, outer unit membrane. What are the features present in outer unit membrane? So this one part is called outer unit membrane. ठीक है, तो आउटर यूनिट मेम्ब्रेन का केमिकल कंपोजिशन है lipoprotein. It is lipoprotein membrane. Its thickness is 75 angstrom. Its thickness is 75 angstrom. And this membrane with specific protein called porin, which are helping transport. So this is what the few features of the outer unit membrane. When you observe or compare with the inner unit membrane, so it's a smooth one, while due to the folding inner membrane is rough. तो ये हमारा होगा smooth unit membrane and completely permeable within a cell and permeable within cell. That means the cytoplasm easily communicate with the mitochondria or mitochondria easily communicate with the cytoplasm. So this is our unit membrane. And as we compare with the next part, that is inner unit membrane. Inner unit membrane. Chemical composition same, lipoprotein, thickness almost same, 75 angstrom, but there is a specific particle on it. यहाँ पर आपको मिलेंगे specific particle या specific molecules यहाँ पे दिखेंगे and these are called oxysome. So due to the presence of these particles over there, this membrane is rough. तो यहाँ पर ये होगी rough unit membrane and it is selectively permeable by nature और nature से होगी ये selectively permeable membrane. So by observing these characteristics of two membrane, we conclude that the two unit membrane of the mitochondria, differ in structure and function. So, the two unit membrane of the mitochondria structurally and functionally differ from each other. दोनों सेम काम नहीं करने वाले हैं. एक ट्रांसपोर्ट का काम करने वाला है बिकॉज ऑफ द पोरिन प्रोटीन और एक ड्यू टू द ऑक्सीसोम काम करेगा सिंथेसिस ऑफ एटीपी में. That's why the two unit membrane are different in structure as well as function. Next, we move towards the next part of your mitochondria, that is chambers. So first of all, the space between two unit membrane is an outer chamber. So firstly, we learn the outer chamber now. कि माइटोकॉन्ड्रिया के आउटर चैम्बर में क्या है. सो आउटर चैम्बर जो माइटोकॉन्ड्रिया का है, यहां पर मुझे क्या मिलेगा. दिस इज अ स्पेस बिटवीन टू यूनिट मेम्ब्रेन्स, स्पेस बिटवीन टू यूनिट मेम्ब्रेन. दो यूनिट मेम्ब्रेन के बीच का स्पेस है. यहां पे कोई भी मेटाबॉलिक रिएक्शन नहीं होती. दिस इज ऑलमोस्ट 100 एंगस्ट्रॉम वाइड. ये होगा हमारा 100 एंगस्ट्रॉम वाइड एरिया एंड इन दिस एरिया नो एंजाइम एक्टिविटी. एंजाइम से रिलेटेड कोई भी एक्टिविटी यहाँ पे होने वाली नहीं है. Why we think about the inner chamber of the mitochondria, कि माइटोकॉन्ड्रिया के इनर चैम्बर में क्या क्या होगा. So first of all, inner chamber is nothing but space enclosed by inner membrane. So when we are talking about inner chamber of the mitochondria, inner chamber, as we firstly think about outer chamber, कि यहाँ पे क्या हो रहा है, आउटर मेम्ब्रेन का नेचर कैसा है वो हमें समझ आ रहा है, इनर मेम्ब्रेन का नेचर कैसा है. नाउ आउटर चैम्बर नो एंजाइम एक्शन, बट व्हेन वी आर टॉकिंग अबाउट द इनर चैम्बर, सो दिस इज स्पेस enclosed by inner membrane. This is the area or space enclosed by inner membrane. Sometimes it is called central cavity of the mitochondria because it is filled with matrix. So this area filled with matrix. यहाँ पर रखा हुआ होगा हमारा सारा मैट्रिक्स और मैट्रिक्स हैविंग नंबर ऑफ मॉलिक्यूल्स. इसके मैट्रिक्स में हमें बहुत सारे मॉलिक्यूल मिलेंगे. पर फर्स्टली दिस मैट्रिक्स इज साइट ऑफ KREBS, Krebs cycle, and it is a part of aerobic respiration. So, the step of aerobic respiration occurs in a mitochondrial matrix is Krebs cycle, and it's a very important pathway of the aerobic respiration. And in this pathway, pyruvic acid is completely oxidized and produce the reducing energy, and this reducing energy converted into ATP by different process. So first of all, the site of Krebs cycle is mitochondrial matrix. This question asked many times in the different examination. So this mitochondrial matrix is nothing but site of aerobic respiration. Now, next we move toward these two parts, Christie and oxysome. What is the importance of Christie and what is the use of oxysome? So these membrane is clear to you. Therefore, I am just erasing these part and move towards the next part. That is Christie and oxysome. Christie. So this area is labeled as Christie. Okay? This area is labeled as Christie or Christa. Okay? So when you observe these Christie, you can easily understand this is nothing but infoldings of the inner membrane. Inner membrane जब fold हो जाती है and projected into the matrix. Okay? This is foldings of the inner membranes. Infoldings. Infoldings of the inner membrane. These are simply finger-like projections. These are finger-like projections and thrown into the matrix or thrown into the inner chamber. So these are the finger-like projections. Number is not specific. But another important point, when they are projected into the matrix, they can easily communicate with the outer chamber because each Christie is having narrow lumen. So why will this easily communicate with the outer chamber? So each Christie with narrow lumen called intracrystal space. So it is not a closed part. It is easily communicated with the outer chamber or inner chamber. So by which the molecule can easily migrate from outer chamber to inner chamber or inner chamber to outer chamber. And this area is just because mitochondria is smaller. So these projections increase the surface area for aerobic respiration. So this increase surface area for aerobic respiration. Aerobic respiration. तो surface area increase हो जाएगा aerobic respiration के लिए. So this is what the utility of Christie or Christa. So infoldings or projections and bearing with a narrow lumen and increase the surface area. So these are the points related with this Christie. Now next, another important part, oxysome. But before it, when we observe the electron microscopic view, कि यहाँ पे पार्टिकल्स अरेंज कैसे होंगे और किस तरीके से ये अपना काम कर सकते हैं. सो इनर मेम्ब्रेन और क्रिस्टी इन अ इलेक्ट्रॉन माइक्रोस्कोपिक व्यू, ये हमें दिखेगा कैसा, ये देखिए. दिस इज अवर आउटर यूनिट मेम्ब्रेन. ठीक है, दिस कंप्लीट एरिया इज आउटर यूनिट मेम्ब्रेन. ठीक है, when we say unit membrane, remember the one thing, this unit membrane is nothing but the lipoprotein membrane or cell membrane. which you observe by the Singer and Nicholson model. So this is outer unit membrane and this is inner unit membrane. And this inner unit membrane shows the folding. This is a folding. So when we say unit membrane, one thing is ultimately clear. This is lipoprotein membrane. तो यहां पर मुझे बाइलेयर लिपिड मिलेगा. ये देखिए बाइलेयर लिपिड और लिपिड के बीच में प्रोटीन. सो दिस इज बाइलेयर लिपिड. तो ये हमारा फॉस्फोलिपिड जो कि हमने सेल मेम्ब्रेन में पढ़ा हुआ. अब बीच में यहां पे कहीं प्रोटीन्स होंगे. दिस एरिया इज स्पेसिफाइड प्रोटीन. So this area is protein and this complete folding is crystal and on it these particles are present. तो ये particles यहाँ पे कैसे रखे होते हैं? There is a presence of one specific complex is there. This is the way. इस तरीके से ये सारी चीजें यहाँ पर arrange होती हैं. So this is arrangement of different complexes. This is called complex 1, complex 2, complex 3, complex 4 and this is complex 5. तो ये जो ऑक्सीसोम पार्टिकल है वो इनर मेम्ब्रेन पे किस तरीके से अटैच है, अभी आप इसको विजुअलाइज आसानी से कर सकते हो. So these are the particles which are arranged on this membrane and all these complexes जितने भी यहाँ पर है ये क्या है हमारे प्रोटीन कॉम्प्लेक्स होते हैं. These are the protein complex. और ये सब हेल्प करते हैं एटीपी सिंथेसिस में. ये सारे कॉम्प्लेक्स एक ही काम करते हैं, हेल्प इन एटीपी सिंथेसिस. As your mitochondria is powerhouse, therefore ATP synthesis is the most important event here. So these all complexes are arranged on the inner membrane of the mitochondria along with the oxysomes. So next part is this one, oxysome. Oxysome. This oxysome is nothing but units of oxidative phosphorylation. यहाँ पर ऑक्सीडेशन होगा और एनर्जी तैयार होगी. सो यूनिट्स ऑफ ऑक्सीडेटिव फॉस्फोराइलेशन और अनदर नेम इज F1 particle or elementary particle. These are also named after the scientist Fernandez-Morgan. So they are also named Fernandez-Morgan subunits or differently named. But this is the most important part. When you observe it, it looks like you divide into 3 parts. So it is tripartite particles. 3 हिस्सों में divide होगा ये. It is tripartite particles. And this tripartite particle is perform the ATP synthesis. So this tripartite particle और oxysome आपको elaborated view में कैसा दिखाई देगा? Christy is clear to you? Now next part है हमारा oxysome. कुछ इस तरीके का आपको दिखाई देगा. Tripartite particle. So 1, 2, 3. 3 part clearly आपको visualize होंगे. This part is called base or F0 subunit. This part is called base or F0 subunit. This subunit just embedded on the inner membrane or crystal. तो यहाँ पर हमें दिखाई देंगे इंटीग्रल मेम्ब्रेनल प्रोटीन और ये है हमारा H+ को स्टोर करने वाला पार्ट. टेम्परेरीली प्रोटॉन्स यहाँ पे स्टोर होने वाले. दिस एरिया इज नेम्ड एज स्टॉक और F5 F6 यूनिट. F5 F6 unit. This area is meant for uphold of headpiece. Headpiece को uphold करने के लिए ये काम करता है. Make in the proper position by which these particles can work easily. So these particles can work easily. This is called headpiece or F1 subunit with a diameter 100 angstrom is the diameter of it. So 100 angstrom के करीब ये वाइड होता है और यहाँ पर हमें मिलते हैं स्पेशली पेरीफेरल मेम्ब्रेनल प्रोटीन. यहाँ पर आपको मिलेंगे पेरीफेरल मेम्ब्रेनल protein, and these protein are along with the one enzyme ATPase. So in the head piece of mitochondria actually ATP are formed, जहां पे actually में ATP बनने वाले हैं. So this is a head piece which actually work for the ATP formation out of them these three parts. So this is all about the elaborated view of your mitochondria कि कहां पर कौन सी प्रोसेस ऑपरेट होगी. सो वी ऑब्जर्व दैट different part having different function. So outer unit membrane, protection, inner unit membrane, increase surface area, right? Inner unit membrane increase surface area. Now matrix perform Krebs cycle. Ly, inner membrane along with oxisome. Inner membrane along with oxisome perform one important step which is known as ETS, electron transport system of the respiration. So these two questions are asked many times, where the Krebs cycle is operate and where the ETS is operate in the aerobic respiration. So different parts come from the different functions. So outer unit membrane, inner unit membrane, matrix, as well as its inner membrane along with oxisome. So these are the structural component of it. Now another important part from the mitochondria, its molecule. कि इसके अंदर कितने मॉलिक्यूल्स मुझे मिलते हैं. तो नेक्स्ट पार्ट है मैट्रिक्स एंड इट्स कंपोनेंट, कि मैट्रिक्स में कितने कंपोनेंट प्रेजेंट होंगे. सो मैट्रिक्स विद इट्स कंपोनेंट्स. सो आई ड्रॉ द स्मॉल स्ट्रक्चर ऑफ माइटोकॉन्ड्रिया अगेन. सो दिस इज योर माइटोकॉन्ड्रिया outer membrane. As you observe along with the projection, it is inner membrane. So usually you can complete the question belongs to diagram. For example, A is indicate which part, B is indicate which part, and that about C is outer chamber and this is D is inner chamber. Okay. So this question is also very common. They can ask either 1, 2, 3, 4 or X, Y, Z or A, B, C, D. Identify the part of mitochondria. Now, inner chamber is filled with matrix. Yes. So this is our part matrix. तो अब हम पढ़ रहे हैं इसका मैट्रिक्स. मैट्रिक्स ठीक है. कहां पर मिलेगा मैट्रिक्स? इनर चैम्बर ऑफ द माइटोकॉन्ड्रिया. इनर चैम्बर ऑफ mitochondria. And this chamber is having number of molecules. Out of them, most important or few molecules I am draw over there. Few molecules. ठीक है. तो DNA, one of the important molecule which make the mitochondria as a semi-autonomous organelle. So this DNA is nothing but double standard circular model. So DNA हमारा double standard circular है. This is DNA. Okay. Now the next molecule is RNA, ribose nucleic acid. जहां DNA वहां RNA. It's one of the common thing. DNA is always along with RNA. So RNA is a single standard molecule. RNA is single standard molecule and it is linear. One another important thing related to the mitochondria, present of ribosomes. Ribosomes. And these ribosomes are like this. इस तरीके के पार्टिकल हैं जिन्हें हम क्या बुला रहे हैं, राइबोसोम. तो each ribosome is made up of two subunit, small subunit and large subunit. So when we just this dimer divide into two parts, so we get a size of this or sedimentation of this ribosome. So it is 70S. And this 70S is made up of the two parts, that is 30S plus 50S, respectively smaller and large subunit. So this is a composition or what we say proto-ribosome present. So in eukaryotic cell, two types of ribosome are present. In a cytoplasm 80S or inside the mitochondria it is 70S. तो माइटोकॉन्ड्रिया के अंदर ये होगा 70S राइबोसोम. So DNA, RNA, ribosome. And the another most important thing is enzymes. Enzymes. As you learn the one sentence from the chapter biomolecule, tertiary structure of the protein is absolutely necessary for the biological process. These are nothing but enzymes. So these enzymes are look like your hollow woolen ball, कुछ इस तरीके के, along with the active site. So these are the enzymes, and point is that these enzymes are perform Krebs cycle. So these are the enzyme for Krebs cycle. So as we learn, the Krebs cycle is the part of aerobic respiration occurs in the mitochondrial matrix because of these enzymes. So this is a cyclic chain reaction completed in 9 steps. So all the enzymes are present in the mitochondrial matrix except one. एक एंजाइम इनर मेम्ब्रेन पे मिलेगा, बाकी सारे एंजाइम्स मुझे कहां पर मिलने वाले हैं, इस माइटोकॉन्ड्रिया के मैट्रिक्स में. So DNA, RNA, ribosome, enzyme. Another one macromolecule presented ATP. As ATP are synthesized within a mitochondria, so ATP are present in the mitochondria. This ATP is first of all called metabolic energy. ATP is metabolic energy and it's a very big molecule made up of three components. First component is centralized place sugar molecule. No doubt it's a pentose sugar, and this pentose sugar is having suppose there is a first carbon of it, first, second, third, fourth, fifth is outside the chain. So here is the presence of one nitrogenous base attached with it, and this nitrogenous base is called adenine, and this is pentose sugar molecule, and its fifth carbon is having 3 phosphate bond. Fifth carbon is having 3 phosphate bond, and these are arranged with the help of 3 bonds. These are the alpha, beta and gamma phosphate bond. So ultimately there is a triphosphate. तो यहाँ पर मेरा आता है phosphate. ये एक क्वेश्चन यहाँ से और बनता है, adenine plus pentose sugar togetherly named as adenosine. They are togetherly named as adenosine, and after that they are phosphorylated. So respectively monophosphate, diphosphate and triphosphate. So mono, di and triphosphate. So this is one other macromolecule present in the mitochondrial matrix, ATP, and few other molecule are present in it. इसके बाद मुझे मिलेंगे कुछ other molecule. Out of them I am right over there the few specific mineral required for the enzyme catalyst, that is Mg2 plus, Ca2 plus and Fe3 plus. These are the 3 important minerals required by the mitochondria. So this is all about the component present in the matrix of the mitochondria: DNA, RNA, ribosome, enzyme, ATP molecule, and few other molecules there. Now, last part of this small organelles is functions of mitochondria. कि mitochondria क्या क्या task perform करता है, क्या क्या functions perform करता है. So functions of the mitochondria. Functions of mitochondria. As you know, status in the cell is powerhouse. So first and most important is synthesis of ATP. And the first most important thing, synthesis of ATP molecule. Synthesis of ATP. And the second function is, there is a synthesis of amino acid. Amino acid भी बना सकता है mitochondria. Synthesis of amino acid. Synthesis of amino acid. Now, regulation of the temperature. Regulation of temperature is done by mitochondria, and regulation of calcium ion is also done by mitochondria. This is all about the 4 most important functions. May you learn the more function in the physiology, but now we are just discussing these much. So this is all about your small organelles present in the cell, mitochondria. In the next video, we are think about the next cell organelles.